Sorry, it will be good in a second. Okay. Just so you guys know, I can only see myself, so if something's going wonky, somebody yell at me. So today we are doing habitat complexity, the effects of habitat complexity on predation rates, and we're doing an experimental demonstration with fire-bellied toads and crickets. So there are several types of predation. There's herbivory, which is animals feeding on plants. We have carnivory, which is animals feeding on other animals. And there's parasitism, which has two main forms. There's non-destructive, which is an organism feeding on another organism without killing them. And then there's destructive, which is an organism feeding on another organism that results in the death of the organism being consumed. An example of this is parasitoids, usually insects, which will lay eggs on other insects, and then the newly hatched young will devour the host. And then there's also cannibalism, and this is a special form of carnivory in which the predator and the prey are the same species. So for prey behavior, in response to the threat of predation, many species have developed strategies to avoid predators. So one of these is aposematic coloration, which is a warning, essentially. So it's a special kind of coloration that advertises an organism's unpalatable or poisonous nature. So many soft-bodied prey items, such as insects, have evolved this mechanism. And the predator only has to encounter the prey once, and then they leave with a nasty taste in their mouth that they won't forget. And for many lepidopterans, this is their primary and only defense, such as the monarch butterfly, which feeds on milkweed as caterpillars, and they store the poison from the plant, and then the adult butterflies actually retain this poison. There's also crypsis and catalepsis. So these are camouflage and the development of frozen postures. And again, insects mainly use this method. So grasshoppers can blend into their surroundings due to their green color, or walking sticks look like the branches of a tree where they live, and they'll freeze when they're approached by a predator. Some vertebrates also use this. For example, zebras' stripes blend into the herd, so the individuals are hard to distinguish. So these are crypsis and catalepsis are good ways to fool some predators, but they're not always 100% effective. And this strategy is also used for hunting. So yellow crab spiders will sit in flowers and ambush predators that come to feed on the flower. And we also have mimicry. So this is highly effective means of avoiding predation that capitalizes on the characteristics of other model organisms. Again, this tactic is common among insects. The non-poisonous visceroid butterfly on the right looks almost exactly like the unpalatable monarch on the left. Also, some types of hoverflies mimic wasps. So if you look like a dangerous or distasteful organism, you can also repel the predators that had bad encounters with these other organisms. Some vertebrates display this strategy. So the king snake mimics the venomous coral snake. And then there's aggression. So some animals have evolved mechanisms to frighten off their predators. These are usually intimidation displays. Examples of this are the large flaps of the frilled lizard, or a toad puffing up to look larger. There's also confusion. So some animals will try to trick their predators into thinking they're already dead. Some small snakes, the hognose and one species of goat on the slide there, have particularly impressive skills for playing possum. The hognose snake will defecate, exude a foul smell, flare out their hood like a cobra, and strike with its mouth closed. So that's aggression. If these aggressive tactics don't work, it just flips over on its back and plays dead. It'll flatten its body, roll out its tongue, and just lie there. Some insects, such as moths, may flash their wings when confronted and reveal a large eye spot. You can see on the slide there. So the flash will confuse the predator, in most cases a bird, into thinking its head is at the opposite side of its body. So the bird will be startled or fly away, or it pecks at the wing, which wounds but doesn't kill the moth. There's also the gray tree frog, which we have here in Sudbury. This will startle its predator when it hops from the camouflaged perch on the tree trunk to reveal bright yellow legs. And also their tadpoles in high predation environments will develop red tails, which confuses dragonfly larvae, which are their predators, to attack the tail instead of their head. So they can live with a damaged tail, but not without a head. There's also chemical defenses. So some animals have developed poisonous or noxious chemicals that they can spray or douse their attackers in to stun them or repel them. Classic example is a skunk or a beetle. There's also polymorphisms. The most frequent is a color polymorphism. This is the occurrence together in the same population of two or more discrete forms of a species in greater proportions that can be maintained by a recurrent mutation alone. So in the specific case of color polymorphisms, the prey will proliferate in the rarer colors, so the predator's search image does not recognize them as a prey item. So we have the example here of the redback salamander. There's a redback morph and there's a leadback morph. Historically, it was thought that the leadback morph was associated with warmer climates, but the more recent hypothesis is that the selection acts on the salamander appearance. And then we have masting. So this is the synchronous production of many progeny by a prey population so as to saturate the environment and satiate the predators. This tactic allows the predators to engorge themselves on prey items. However, such a large number of offspring are released that the predator can't eat them all. So some percentage of the progeny will survive. Cicadas do this and oysters. So just as prey species have evolved mechanisms to cope with predation, predators have also evolved ways in which to deal with these mechanisms and attempt to overcome them. So natural selection is generated by competition between predators for food. Predators have thus incorporated a variety of strategies that allow them to capitalize on their prey species. So for feeding strategies, when to feed, the time of day is an important variable for many predators' feeding strategies. For example, many herbivores have adopted a night feeding strategy to avoid predation for themselves. There's also mobile predators, so this can be hunting. It's effective but an energy expensive method of prey capture. The fastest and strongest predators, for example, wolves or cheetahs, will run down and kill the oldest or weakest members of their prey species. And this helps create a healthy balance in the prey population by eliminating the sick and debilitated individuals. There's also sedentary prey. So predators can browse on sedentary prey, such as colonial organisms or plants or algae. So predators will harvest some of the colony and may even cultivate it so that some of the prey species is left alive. And this method results in a high success rate, requires little energy, and these predators are generally more successful than those who actively hunt. However, grazing can expose the predator so that it becomes prey for another species. There are also stationary predators. So you can be a stationary predator that feeds on mobile prey. So sea anemones wait for the food items to brush against their stinging tentacles. And then they discharge their stinging nematocysts into the prey item, paralyzing it. Even some plants have evolved into stationary predators. For example, the Venus flytrap and pitcher plants lure insects in, trap them, and digest them. Now, there are many factors that can affect a predator's ability to catch prey. These can be both biotic and abiotic. The first one is search image. A predator may have a particular preference or look for one particular color form of its prey. Tinbergen termed this prey form the commoner. This allows individuals of the prey species to use camouflage or polymorphisms to protect themselves. And many nectivores have a search image that consists of one particular species or color flower. There's also landmark learning. A predator may familiarize itself with a particular area where prey are abundant. And there's habitat complexity. So prey can seek shelter within the habitat. So as habitat complexity increases, it becomes harder for predators to find prey. This is the idea that we're going to be testing in the lab today. There's also environmental stochasticity, which are random chance events or variables present in the environment that could affect the balance of predator and prey. There's physical variables such as salinity or fluctuations in temperature. And then there's also weather, which are seasonal changes and sudden rapid changes as the result of storms and hurricanes can also drastically affect the balance of predator and prey. So today we are doing the hide and seek predator prey lab, looking at habitat complexity. So the purpose of this experiment is to examine predator-prey interactions in habitats of varying complexity. So fire-bellied toads will represent the predators and crickets will represent the prey. So we're going to test 3 habitats that range in complexity. We're going to have the same gravel across all 3 and then the addition of 1 or more layers of artificial plants that allow the crickets to hide. And normally we would do this in lab in the person and we test 6 habitats. But this year we have provided you with a video demonstrating the experimental setup and procedure, and we're also providing you with the data set to be used for your final lab manuscript. So we have 3 habitat treatments. The first one is the simplest. So natural gravel to provide color camouflage. This is low habitat complexity. Then we have moderate complexity, natural gravel with 1 layer of plastic plants. So medium complexity. And the most complex is natural gravel with 2 layers of plants of varying heights. So our objectives is to determine if habitat complexity affects predation rate. So you're going to be, the video recorded the kill times, and then it's going to be graphed as predation curves. And the graphs will be compared over the different habitats. And then the slopes and the shapes of the curves will be discussed. And we're going to be making predictions for all 3 habitat treatments. So you guys were provided with this worksheet. So we're going to be talking about this in the breakout rooms about developing an hypothesis and predictions. So your hypothesis is the why, it's the explanation, and then your predictions are what you think is going to happen. So for our procedure, this is what you're going to be seeing in the video, but just a brief overview of what's happening. 10 crickets will be introduced into the habitat, and we let them acclimate and seek out refuge for 10 minutes. And then 3 toads are added, so that's time equals 0. And then we record when a cricket is eaten by any toad. And then the timing is stopped after all crickets are eaten or after 1 hour has passed. So you're going to be graphing a kill curve for each of the 3 habitats tested and showing all 3 treatments on one set of axes. So you're going to plot time until kill on the x-axis, which is your independent variable. And you're going to plot number of prey eaten on the y-axis, which is your dependent variable, and you're going to connect the points with a smooth curve. And we'll probably discuss this more next week, but did the outcome support your hypothesis, and does the data match your predictions? So your final manuscript will include your kill curves made by hand or using Excel. I would probably recommend Excel because it gives you the R-squared and the equations automatically. So it's probably just a little bit easier. So the data set includes 3 replicates of each of the 3 habitat treatments. So you're going to be plotting the mean data, and you can fit the trend lines in Excel. And again, it gives you the R-squared and equations. And make sure you describe your habitats in your lab manuscript, which is due on November 30th. And of course, review your notes from Lab 1 about how to write a scientific manuscript, and look at the feedback provided by your GTAs on all your previous assignments. One of the main outcomes of this lab is to hone your scientific writing skills, which is why we've been doing this in pieces, and now it's culminated to the final manuscript. So just briefly, this is your marking rubric for the manuscript. We'll probably go over this again next week just to really drill it in. But the introduction, we're starting off broad. We're doing this hourglass shape that we talked about in Lab 1. So you start with your background on the subject, then you give your justification and purpose for your experiment. Then you get narrower into your hypothesis and predictions. This can all be done in 3 paragraphs. Your methods are in past tense. You're going to describe the experimental setup, all the details of the procedure, and always use full sentences. Don't give me a grocery list of what happened. For our results, this is where we have our tables or figures with proper captions and axes, like you've done before. And you're going to give summary sentences of your results, and don't be redundant between your figures and your text. Your discussion, so we're starting narrow. Your hypothesis, was it supported or rejected? Why do you think that? A hypothesis never proven true. Your data either supports it or does not support it. And then you start going out broad again, and you compare to what other people have found. And then you conclude with why your results are important, and maybe give some future directions for research. And for your literature cited, we're looking for a minimum of 6 papers from the primary literature. So that's scientific articles. So go on Google Scholar. That's what you need. So Lab 8 next week, we're going to discuss more details about the prep of the manuscript. You're going to bring us your hypothesis, predictions, and some draft kill curves. You're going to bring us a bulleted outline of your manuscript, and you're going to bring citations for at least 6 papers relevant to this experiment. Okay, so that's it for the pre-lab. We're going to watch the video now, and then we'll go into our breakout rooms to talk about hypotheses and predictions. Just give me a second to get the video up. I'm going to stop my video just so the video hopefully runs better. Where did my... Okay, 1 second. Okay. Sorry guys, bear with me. Okay, and if it's not working properly, somebody let me know. I'm going to start it now, and hopefully it runs smoothly.
Welcome to the Hide-and-Seek Predatory Prey Lab.
Hello everyone, welcome to the Animal Ecology Lab, in which we're going to be looking at the effects of habitat complexity on predation rate. Our predators in today's experiment will be fire-bellied toads. That's these guys here. They are easily available in the pet store, and that's where we obtain these guys. So they will serve as our predators today. Our prey are going to be crickets that are also easily available from pet stores. These are the classic guys that are fed to pets. You might have a leopard gecko or a crested gecko or something that you feed at home, and these are the same crickets for that. And we'll be testing the impacts of different habitat complexities on predation rate, so on the kill rate, how quickly these toads are able to find and eat the crickets in the various habitats. We have 3 habitat types that we'll be comparing. One is a very simple habitat type. So all it has is gravel of a natural color and nothing otherwise that the crickets can hide in. It does provide some level of camouflage because the color is similar to that of the cricket bodies. The second habitat type is a little bit more complex. So we have the same substrate, natural colored gravel, but then we've added one layer of low plants. So this will provide the crickets some areas to perhaps hide from the predators. And the third habitat type, this one is the most complex. So it has, again, the same gravel type, but now we've added 2 layers of plants. So there's 3 low plants and 3 high plants. And so you can imagine that there's lots of places for crickets to hide in this particular site. Hi again, everybody. So one of the things that I want you to think about and want you to talk with your GTAs about during the Zoom session is what your hypothesis would be. So what your explanation for differences in kill rates might be amongst these habitats. And specifically, I want you to make a prediction for what you expect to see for kill rate in each habitat. I want you to think about a rational explanation for why there might be differences among the habitats. And I've already given you a few hints related to the plant layers and where crickets can hide. So I want you to think about how predation rate might differ, but then if you're graphing it, which you will have to do, to also think about how the shape of a curve might differ between the different habitats. Can crickets be found a lot faster at the initial part of the experiment, and maybe there's more hiding later? But just think about those kinds of things as you're thinking about what you will predict for each experiment. Okay, so now we're going to go over the experimental procedure. So the first thing that happens, we're going to start with the simple habitat. The first thing we do is introduce the 10 crickets into the habitat. And we give them 10 minutes to walk around and get used to the habitat. Okay, so now 10 minutes has passed. So now it's time to introduce our predators, our beautiful 3 fire-bellied toads, who, if I can get the lid off, will serve as basically a super predator. So we're considering all 3 toads as sort of one big predator in this experiment. I'll take this chance also just to show you how beautiful they are. So that's bright green on the background, bright green background, and then they have these beautiful colorful bellies. So I need to introduce them all quickly, get the lid on, and now the experiment begins. So this is time 0 for the start of the experiment. And then you need to record, there's a kill. Did you see that toad? He just ate cricket. So that's the first kill. So then we recorded the time that first kill happened. There's a cricket on that toad's back. We'll see if anybody notices. Oh, kill number 2. This toad just ate a second cricket. And we'll keep watching and recording the time that each one of these kills is happening. This is how we will calculate our kill rates or our predation rates for the experiment. There's another kill, a successful hit, and that's recorded then, again, the time at which that happened. Another one was just killed and eaten. Another one over here, looks like that was a miss, so we do not count that. Nope, he got it. So we count the time for that one. I'm trying not to point my fingers as they're doing it because I don't want them to look at my fingers instead of the crickets. Oh, so that was a hit.
No, he got it.
He got it, There we go. So that's another kill, and again, somebody's standing on his head. And so we continue recording each kill until all 10 crickets have been consumed or until 1 hour has passed. So we would continue to record the data, noting all the hits, all the kills for each cricket until, again, all 10 are consumed or 1 hour has passed. So we are still progressing in the experiment, testing the simplest habitat here. And one of the things I want to point out is that the only way the crickets can escape notice in this case is to use camouflage and catalepsis, that being still. So we've noticed, actually, as we've been watching the experiment, that the crickets have been going into freeze postures. And that makes it really hard for the toads to find them because the toads are visual predators and they need the crickets to move to find them. So it's interesting, there are still 1, 2, 3, 4, 5, 6 crickets left, it looks like. Maybe. And if they stay still, it's really hard for the toads to see them. So that's also something to keep in mind as you're thinking about your predictions for the experiment, is the behavior of the prey as well. Okay, so now we're going to do the procedure for the medium complexity habitat. So remember, this is the one that has a layer of gravel, which is the same color as the crickets, and has 1 layer of low plants. So the first part of the setup is to introduce the 10 crickets to the habitat. We give them 10 minutes to acclimate and try to find some places to hide. Okay, so our 10 minutes has passed, and now it's time to add our predators. So again, we have 3 fire-bellied toads. Going to try to give them into the habitat all at once so that timing will start right away with all 3 toads. So here we go. begin timing, recording all the hits as they happen. Now remember, the crickets now have had some time to hide and get up in the plants and hopefully be able to escape.
Oh, there's his first kill.
Looks like, did he get it? Or did he just notice it?
Well, he struck at it.
He struck at it but missed. So that has to be a successful kill to count in our kill curve. And there's one, first one. So see, it took a little bit longer compared to the simple habitat for that first kill to happen.
They're very much like the kamikaze ones that go in the back.
Much more time is passing before the second hit is happening, second kill. And there's a second successful hit. All right, so now we're going to show you the setup for the most complex habitat. So this one, again, it has the same gravel, has the color camouflage gravel, but now we have 2 layers of plants. So there's 6 short plants and 6 tall plants. So lots of places now for crickets to go hide. So the first thing, of course, is we introduce the crickets into the habitat and we give them 10 minutes to acclimate and to find hiding spots. Okay, so our 10 minutes has passed and the crickets have had some time to find places to hide. So now we're going to introduce our fire-bellied toad predators again. And I'm going to introduce all 3 at once again. I'm going to put them into the tank. And now the time starts immediately at the introduction of the toads. And so think about what might happen in this case, because now the crickets have lots of places to go. And the toads need to maybe search a little bit more to find those crickets. And think about the comparison of how things happened in the simple habitat and how things happened in the medium complexity habitat, and now how things might happen in this most complex habitat.
There's a great shot of the underbelly.
So I can see crickets are actually going up into the vegetation, getting high, which will maybe make them less obvious to the toads. We also have a toad who is way up high in the canopy of this forest. Maybe he'll be the one to feed up high. Did he get one? From your side, could you see?
Fighting. They want to be on camera.
The other thing to think about too when we do this experiment in person is as humans, we try not to hover completely over top of the setups, because the toads might then think we're predators and they might actually be a bit frightened of our presence. So when we have done this experiment as a lab group in person, we also try to not sit really over top of the tank and not look scary to the toads. That toad seems more intent on getting out than eating. Oh, 2 of them. Okay, so the first hour has passed now and the experiment is completed. So we'll do a quick comparison across the 3 different habitats. So again, starting with the simple habitat, you can see some fat frog bellies, fat toad bellies in here, and there are no crickets left in the enclosure. So all 10 crickets have been consumed. When we move to the medium complexity habitat, you can see that there are still some crickets wandering around. Here's a cricket here at the front, and there's still a couple of crickets that are there because they were able to find refuge and hide. And then in the final habitat, the most complex habitat, there are still several crickets left. Only a few have actually been consumed because most of them have been able to find good hiding spots. So again, I want you to think about why that would be and try to explain that when you're creating your hypothesis and your predictions for each one of these. Part of the exercise that you will have to do with these data that we are providing for you is to graph the kill curves for each of the different habitat treatments. So we've provided you with an Excel spreadsheet that has the data, and the data include 3 replicates of each habitat treatment, and then there's the mean data calculated for the time it takes for each kill to happen. So you're going to have to include a graph in your manuscript that you hand in in 2 weeks from now. And so on the x-axis will be the time to kill, and remember to each kill, so each cricket being eaten. And this duration of the experiment is 60 minutes. So you'll have 60 minutes on that x-axis. And on the y-axis is the number of crickets eaten. So it's a cumulative curve of number of crickets eaten. And this is why you had to record, why we recorded, the time that each cricket was consumed. So then what you'll do is you'll plot each cricket kill. So this is the first cricket, so cricket 1, cricket 2, cricket 3, all the way up to cricket 10. And remember, in some cases, not all of the crickets were actually eaten. So you will create your curves based on that. So for example, in the simple habitat where we had all 10 crickets eaten, you will plot those data. So say the first cricket was consumed at 1 minute. So cricket number 1 consumed at 1 minute. Cricket number 2 was also eaten quite quickly after, if you remember. So maybe it was just a couple of minutes later. And so then you will plot all of the data, and you may end up with a curve that looks something like this to represent the simple habitat, the most low complexity habitat. And then similarly, if you remember for the most complex habitat, the first, I don't even know if we recorded a first kill in the time that we were there. So, you know, it took quite a while. The first kill, maybe it happened at 30 minutes later. So your first data point is actually there. And then maybe only a few crickets ended up getting eaten. And so the shape of your curve is going to be quite different, perhaps. So use the data that we've provided and create this kill curve. And this is part of the assignment that you will hand in with your manuscript in 2 weeks from now.
Okay, so that was your video. That was the experiment. Pretty cool to watch. So yeah, the only thing left to do now is to go into our breakout rooms.
Jackie, do you think
Okay, so it looks like we finally got the breakout room thing to work out, so that's really good. I guess we can start with, does anybody have any questions? I know there are some questions put in the chat and answered. So does anybody have any questions about how this is all working?
I have one for the final manuscript.
Yep.
For the kill curve that she was just talking about, the graph, do you want them all, like all 3 of the like curves on one graph, or do you want them separately?
All 3 of them go on one graph.
Okay.
So I think it says that somewhere in one of the handouts, but yeah, all 3 of them are supposed to go on one graph, which can be a bit of a pain to do sometimes. I know some students in the past have struggled with it, but give it a go. Try your best. But yeah, they should all be on one.
But we can also, wouldn't it be easier if we made it by our hand? Like I read in one of the handouts that it said use your hand. So if you're making 3 graphs, the 3 kill curves on one graph by hand, wouldn't that be easier compared to making it on Excel?
It could be, but the only problem is when you want to make the kill curve, they want you guys to put, well, we, I guess, want you guys to put the R squared value. And if you do it in Excel, you can just like right click and say like include R square value. Otherwise you have to calculate it by hand. So I would highly recommend doing it in Excel. It makes it neater and it will probably be easier for you in terms of the math.
Sounds good. Thanks.
Yep.
Do you want the lines all in blacks with different symbols, or do you want grays as well?
Blacks would be preferable. That's one thing that I wanted to mention. I have a couple notes, so I'll leave it open if anybody has any questions, but I'm almost done marking your results and your discussions, and I have a couple notes just to like give you guys a heads up. I'm hoping they'll be done getting marked by the end of tomorrow, so you guys should have them by then, but it might be into Wednesday or Thursday, just depending on how schedules work out. I've made all the notes on them. I just have to like actually mark you guys, and I take my time trying to make sure that like I'm being as objective as possible and docking people the same if they make similar mistakes. So with it being a longer submission, it takes a little bit longer to mark. But like I said, I'm hoping it'll be done by the end of the day tomorrow. And you guys should have all your feedback and everything. So are there any other questions about the experiment and how it all works and all that?
Is that video going to be available for us to rewatch later if we want to?
I don't think so. It's currently not posted on D2L. We may rewatch it next week during the pre-lab. There is an option in the slides to rewatch it if we feel it'll be beneficial to the discussion, but I don't think that the video will be available for you guys to watch whenever you want.
Okay.
But all the information should be in the handouts and everything, so you should, if you pay attention to the video, you should be able to discern anything. And of course, if you guys have any questions about what the setups are like or anything, you can always feel free to email us and we can try to help you out with that. Any other questions? Okay, if anyone has any questions, you can always put them in the chat. I know sometimes they occur to you later on. So in terms of your results, overall, they were pretty well done. A couple things that I wanted to mention. So when you are making your results in your discussion, and I know that I said this before, but I think it might have been misinterpreted by a couple of people just based on what I saw. So your results are supposed to just be what happened, right? So like you'll have just statements of, we found this, it had this significance, this is what we found. End of story. And then your discussion is kind of what goes into, okay, well, maybe this could explain why we found what we found, or this supports our hypothesis, or this refutes our hypothesis, and all of that. So generally that was pretty well followed. A couple people did put a little bit of explanation to the results that shouldn't have been there, or a couple statements that might have been a little bit iffy. There are also people that I know it is difficult when you're writing it in pieces, but there were people that included things in both the results and the discussion that would typically be part of your materials and methods. It's tricky when you're only writing the results because you want to include all the information, but if you had been writing the entire manuscript, it would have been redundant. You would have already explained what equation you used. You would have already explained all of that in the materials and methods. So that being said, though, with the results, while it is just what you found, you still need to have text in your results. So there was a couple of students that had just a table or a couple tables in their results. You still have to have text introducing the tables and stating some of your results that might not be like, what is the word? Not directly, but so stating some of your results that might not directly be in the table, right? So, and that goes hand in hand with your tables need to be introduced before you present them. So typically you would have something like, if we're doing like the popcorn lab, just because that's what you guys wrote it on. So you'd be like, okay, so the average number of captures during our experiment with the first methodology was blah, blah, blah. And then you'd put table 1 in brackets, and then you'd have table 1 there. And then you would go on after table 1 to say the average number of recaptures during the second methodology was X, with a variance of whatever, and then you put table 2. So that way, if you're introducing each one of your tables, you're addressing each one by itself, so it makes it less complicated, and it helps to like break up your results in a logical way that's easy to follow and read. Does anybody not understand what I mean by that? again, if you have any questions that you don't want to like say in front of the group, you can always private message me in the chat, or you can send me an email. But that was a big thing, is you have to introduce your tables and figures before you present them, and try to present them in order and separate from one another. Because if you have a whole paragraph about all your tables and then you have all your tables, it can get a little bit confusing because you're going back and forth. And if you have anything that is like a comparison, so say if you did do some statistical comparison to say, yeah, we got significantly more recaptures in the first methodology than the second one, you would put that at the end, because you already explained the results you found in each one, and then you can explain the results between the two. Hopefully that makes sense. I put more detailed notes on each one of your submissions. So if you did do that, hopefully those notes will clarify. Again, if you have any questions or you think something was marked weird or anything, please let me know, and I will be more than happy to try to either explain it or look into it to see what happened. The only other thing, references. Not very many of you, but a couple of students in the actual literature cited, so like the not in text part where you actually have like all your citations, they put like the first author and then et al. You can't do that. So I didn't like really nail you guys, because I don't know if it was explicitly explained, but you can put et al. In the in-text citations, but when you get to the end, you have to put at least the first 6 authors. I don't think there's any citation style that you can put less than 6 authors, and usually it's like you put the first 6 and then ... and then the last one, and that's if there's a whole bunch of authors, which some papers have like 20 or 30. Sorry, there's a fly. So that's important. Also be aware, because I noticed, I don't know if it was intentional or unintentional, but I do check to make sure that you actually used all of your references in the text. So there's a couple people that had a citation at the end in their literature cited for a paper that you never actually referenced in the text. And I do check for that, and I do notice it. So I don't know if it's accidental or if it's intentional. If it's intentional, don't do that. And if it's accidental, make sure you double check, because I know sometimes if you write a sentence and then decide you're not going to use that reference, you might take it out and forget to take it out of your lit cited. But double check that, because I will be checking for it, and I will have to dock marks if it doesn't match up. Is there anything else? Oh yes, and in your discussion, a lot of you did it, but a lot of you didn't seem to do it. So you guys remember how we talked about that like hourglass shape of what your paper is supposed to look like, right? So in your introduction, you start off really broad, and then you say like, here's the general field that we're in, here's some like knowledge gaps, and here's what I'm going to do to address these knowledge gaps, and you're in your middle. And then same with like your materials and methods is very specific to what you're doing, and your results is exactly like very specific to what you're doing. So in your discussion, you start to come out from that hourglass. So you start off in your discussion really talking about your results. So some people kind of, once you got into your discussion, you started off broad again and started talking about like population ecology as a whole. And that's not, you're supposed to start off with kind of contextualizing your results and comparing it to other literature, and then kind of like broadening it out to what does this mean for the whole like discipline, what you found, what does that mean for the whole like field of study that you're in, and then you can relate it back. But some people, again, I know it's complicated because you're doing bits and pieces, but some of the stuff like that would have been a perfect paragraph for your introduction, but it doesn't belong in your discussion. So again, once you guys read your notes, hopefully that'll be a little more clear, but those were just some of the bigger things that I noticed. And I kind of wanted to give you guys a heads up in case any of you are starting to write over the next week, if you're starting to write before I actually get your notes back to you, of just some of the more common things. In terms of the graph, so you guys were asking about shades of gray. Technically, it's all supposed to be black and white. No color, no shades of gray. Everything is black and white. If you want to discern between your 2 lines, if you have like markers, you would have like the first line could have like filled in triangle markers, and the next line could have filled in circle markers, and then the bottom one could have squares, and you could have non-filled markers, or you could have like markers with stripes in them and stuff like that. This is it's a little bit outdated, I know, especially because a lot of publications are moving to electronic forms. But so in electronic forms, it doesn't cost you any more money to print in color. And so you can use more color in photos and stuff. That being said, while you guys, so I know you guys aren't actually doing the experiment, but if you were including photos, I would accept color photos for this, because most of the time people do include photos. In the past, it's going to be almost impossible for you guys to, because you didn't actually do the experiment. But yeah, graphs and tables are all supposed to be completely black and white, no gray, no color, no nothing, just because it would cost more to print them when they were actually like in print. And I think that's all that I wanted to say about that, I think. Does anybody have any questions about anything that I just said, or concerns or anything like that?
For the graphs, can you also have a dotted line?
What do you
Like
mean?
you have so you can have markers like a circle or a triangle or a square to represent like which line is representing what data. But let's say like you have so many lines and you can only have so many icons on to act as markers. Could you make some of the lines themselves like a dotted line, or so it's not just a completely filled out line? You know what I mean?
Yeah. So you can. Typically, so I don't think you guys will have to resort to that for this, because you only have 3 lines. So you should be able to use markers. People do use dotted lines, especially when you have graphs that have like a whole whack of lines. But usually the way that they do it is they like group things together. So how do I explain this? So it doesn't really make sense, but I'm going to reference, so like your popcorn lab, right? You had 3 different methods. So if you were graphing your like several things in each method, you could have like long dashed lines for the first method, fine dotted lines for the second method, and then solid lines for the third. You know what I mean? So you can use different line types, but it's usually if you have like 9, 10, 11 plus lines. You guys should be fine with just using the different markers, but there's nothing like technically wrong with using dotted lines. The only thing is it can make things a little bit like unclear depending on what pattern you choose. If it has like really wide gaps and stuff, it can be a little bit weird, but there's nothing technically wrong, I don't think, with it. But I would recommend using markers and solid lines if you can. Any other questions?
So Heather, I'm looking at the fabricated data for this experiment, and for when the habitat complexity is high, the last 2 kills, kill 6 and 7, they have no mean, which obviously means that the toads they did not kill within the 1 hour time for the kill 6 and 7, right?
Mm-hmm.
So
I think so. I can check, but keep going. I'm going to bring up the thing while you're talking so I can double check.
Yeah, but basically my question is, because for kill 6 and 7 for the high habitat complexity, we don't have any data for any data. So do we still plot them, or do we just use 0, or do we only go as far as kill 5?
So you're saying that basically like kill 6 and 7 happened after the experimental time?
Yeah, after 1 hour's time, right?
Yeah,
because
so you wouldn't plot those because the experiment ends at 1 hour time. You can mention in your like results, and you can talk about it in your discussion that 2 of the crickets didn't get killed within that time frame, but if any of them get killed outside of the time frame, it's technically outside of the experiment.
Okay. Yeah, that answers.
I believe so. I can double check with Dr. Litskis. I wonder if I can, because I had a little option before to call her in. Do I still have that? I don't think I do. Yeah, I'll double check for you on that. I'll just write that down.
Thanks.
But I'm pretty sure you don't have to graph them. She'll probably come and check in on us anyways, and if not, I'll email her or I'll talk to her after the lab and then she can clarify. Any other questions?
Are we doing anything else in the breakout room?
Yes, we are going to be discussing all of your hypotheses and predictions. I know it's weird because we already watched the video and you already had the data, but that's one thing that you guys have to bring next week so we can talk about like hypotheses and predictions as a whole. I just want to make sure that there's no questions about the experiment or about your results and everything so far. And if there aren't, then we can move on. Okay, I'll take that as there's none. And again, if you have any questions that come up, feel free to email me. So in terms of our hypotheses and predictions, can somebody explain to me the difference between a hypothesis and a prediction?
A hypothesis is testable.
Yes. Well, your predictions, I guess, are sort of testable too. So your hypothesis essentially gives an explanation of why. So a lot of people use if and then statements. I don't know if Litzkis has talked to you guys about that. Anyone recognize that if and then statements?
Sorry, I was just taking a drink. We have talked a little bit about it, but not really using like if and then.
Okay, so really what I find the easiest way to talk about a hypothesis. So your hypothesis, like I said, it explains why. So lots of people use if and then statements as in, if this is true, then we expect to see this. So it kind of like ties them together. So your predictions are just purely, what do you expect is going to happen? What do you think you're going to see? You just say, like, you could have your predictions be all sorts of things, and you don't really give an explanation as to why. Your hypothesis incorporates that. So if you're talking about the toads, can anyone give me an example of with your toads? Like, does anyone have a hypothesis kind of? Like, what do you think is going to happen and why? And then we can try to formulate it into more of a sentence, right?
Hi. So I hypothesize that the crickets in a natural gravel habitat will have a higher predation rate from the frogs just because they have less of a area to hide in.
So you mean just like the plain habitat?
Yeah, plain gravel.
Yeah. Sorry, I'm getting it a little bit confused in my head because I don't know if you guys read the word or like the handout, but when we originally did the experiment, so you guys are only working with 3 habitat types, right? So you have like your natural gravel, just plain natural gravel with some plants, and then natural gravel with lots of plants. Those are essentially your 3 treatments. When we do it in the lab, historically, we've had those 3 treatments, but you do the same thing with it's like a blue colored gravel, so the crickets don't blend into the gravel. So I was just like, I got a little bit confused. So in just the plain, yeah, I would totally agree with you that that's probably what's going to happen and why. So if you want to phrase that as an if and then statement, you could say like, how do I put it? If the crickets are able, or if the vegetation provides the crickets with more places to hide, then we expect that the toads will have a lower predation rate in more complex habitats. Right? Do you get what I mean? Like, it's more general. You gave me one for just that, but that's more of a general hypothesis, right?
Yeah, no, I completely understand.
Yeah. And like, there's nothing wrong with having hypotheses for each habitat. Especially if you think that something vastly different is going to take place in one or the other. But yeah, your prediction, so based on what you said, your prediction is that they're going to eat the crickets faster in the plain habitat, like without any vegetation. They're going to be able to see them and they're going to eat them faster, which is perfectly logical. And as you know, is kind of how it turned out. So it's not as fun to do predictions when we already know what happened. But does everybody like feel clear on that? Because you're going to have to give, I think the handout, you have to provide predictions for each habitat, and then you have to give an overall hypothesis, which is kind of what I just like gave you. Try to switch it up a little bit if you can. Where's the handout? So you guys have to bring that in for next week. Just be aware of that. And you have to bring your draft kill curves, and you have to bring 6 citations. And we're going to be kind of talking about the citations. Dr. Litzka said she's totally like, you guys shouldn't be working together on things really, but like if you guys are sharing citations, she's totally okay with that, because it's all out there. It's all your data sources, and you guys can interpret them in different ways. But yeah, we're going to share some of the citations. So yeah, your hypothesis is like your overall hypothesis, and then you have predictions for each one and rationale for each one. So you're kind of making a hypothesis for each one and one overall.
So we aren't submitting these items next week. We're just bringing them with us to lab.
Yeah, we're bringing them with you to the lab, and then we can kind of like talk about them a little bit more. And we're going to go over more like in detail what we expect from you guys in terms of like what we're going to be marking. And just to make sure everybody's like on the same page and that we're all making sense of what's happening. It's so definitely, and it's kind of to make you do some of the stuff now so you're not leaving it all to the last minute. So is there any like questions or confusion about that? And I'm sorry you guys have to watch a video instead of being able to watch the toads. But I gotta tell you, once you've been sitting there for an hour, the toads are cool, but watching them eat crickets for an hour can be a little bit daunting as well. So yeah, other than that, definitely I would get started as soon as possible on your stuff. Make sure that you read all of the notes that were given to you on your previous submissions, because I gave a lot of notes, and it's very, very obvious to me that some students are not reading said notes, or at least don't care, because you're making the exact same mistake over and over and over again. Especially, it's very disheartening with the tables. I mean, some people, like if it's a minor mistake, I get it, people screw up, but some people, like I said, you guys have to put effort into, like I try to provide as detailed notes as I can. And if any of them are confusing at all, please email me, because I'm doing it for your benefit. Like it doesn't really matter to me if you leave this class without being able to make a table, but it'll impact your mark negatively. And I'm trying to give you guys everything you need to succeed. And please feel free, if you have any questions before you submit things, to email me. I've had a couple students and I've tried to help them as much as I can. But definitely look at your notes and review the first PowerPoint, because the things that are listed in the PowerPoint are the things that we are specifically looking for. Yeah, and I think other than that, I think that's pretty much it, unless anybody has any questions. I can just double check to make sure I'm not missing anything. So yeah, bring your hypothesis, prediction, draft kill curves, bring a bulleted outline of your manuscript. I forgot that part. And bring citations for at least 6 papers relevant to the experiment.
For the kill curves, did you want us to graph the mean or all 3 replicates?
I'm not sure exactly what you mean.
Like for each treatment, there's 3 replicates, right? And then a mean was provided to us. Like, did you want us to graph the kill curve for the mean or each of the replicates?
I believe it should be for the mean, because is it a mean for, okay, wait, let me just double check here. I apologize for any confusion. Again, this is not how it usually is run. Usually it's run in the lab and then you guys get all the data. Let me just take a look at data. Okay, yeah. So you have a mean for like what time each kill happened at. So I would say that you should be graphing the mean. So you should end up with a graph that has 3 lines on it. We can double check with Dr. Litzkis, but I feel very confident in that. Are there any other questions? So I got a message from a student. So I predicted that there would be a negative correlation between habitat complexity and predation rate. So yeah, not sure if it's worded correctly. So if you have a negative correlation between habitat complexity and predation rate, it would mean that, because your habitat complexity would be here, I'll draw it out. A little bit of an easier way to like think about things. So as you guys know, whenever you're doing experiments and stuff, you usually have your independent variable and your dependent, right? So your independent variable is independent, as it says. So usually it's things like time, right? Because time goes on no matter what is happening. So if you're thinking about how correlations go, so I hope you can see it. So yeah, your dependent would go on your y-axis and your independent goes on the x typically. So if it was like predation over time, predation is on your y and time would be on your x. So if you want if you're saying that there's a negative correlation between habitat complexity, which would be your independent, so independent is habitat, and predation would be dependent. So if you're saying it's negative, then that would mean that as your habitat gets more complex, then your predation would go down. So yes, that is worded correctly. But if you're ever like concerned that whether you're wording it correctly, I would recommend drawing that out, drawing out your graph, figuring out which one's dependent, which one's independent, and negative is going down, right? So yes, that would be how it would be worded correctly. Any other questions or anything? No? Okay, I think that that's pretty much it then. I would definitely take advantage of this time to start. Again, I hope to get your notes back to you as soon as possible. And yeah, so otherwise, everybody, you can go back to the main room, I guess. I don't think there's anything else.
No, have you tried with your family, Jackie, Jackbox?
No.
It's such. I don't know if you guys have tried that yet. It's an online. It's a game you can play with anybody, and all they need is a computer wherever they are and their cell phone. And so you draw things on your phone, and you have to guess it. You have to guess who drew it or what it's supposed to be a drawing of. It's kind of like a version of a lot of different games brought together. But it's real time. So you're talking to them like this, and then we'd be guessing at, and somebody would write something like, what does this word mean, right? And you'd have to explain it. And what you're trying to do is get someone to take, guess your picture or guess your.
So it's like Cranium. It's like a live version of Cranium.
Yeah, some of the same kind of ideas with Cranium, but it's real time on your phone, on your device. So it's like a board game, but it's so much fun. We played a lot during this.
Yeah, me and my friends played that like all the time during like March when everything was like shut down. That's what we did.
What's it called? Jackpot?
Jackbox. A lot of fun.
Jackbox.
Just remember, there is a adult and a child. There's a difference. At Saber's birthday party, I ran it. I didn't know there was a kid one at that time, so I ran it, and oh my god, I can't remember what it was, but Ernst texted me and said, "Hey, we got a question about like it was." I can't even remember. I don't want to make it up, but it was like an inappropriate term, right? And it was the actual question was, what? Give another example of this, right? And the kids, all the boys that were at the party had no idea what the word was. So Alice and Ernst texted me both, said, "Hey, Odd, Leo asked what this was today." I was like,
oh no.
They're like, "We thought we'd set up a Zoom meeting and you could explain it to them."
Okay. So Charlie, Taylor, Zoe, and Luis, you guys, if you're finished, you're free to leave. That's okay. So those they're all from Heather's room. Yeah. Yes, that would be our preference. And so Steph's going to make a video of the graph exercise and post it on D2L when her computer stops getting angry. Yes. So not necessarily a linear line. So that's why it's very convenient to do it in Excel, because Excel, you can pick a trend line to fit to the data, and you can try a few different trend lines and see which one has the highest R squared value. No. So you have 3 replicates of each of the 3 treatments. So there will be 3 lines on your graph. Each line will be the mean for one treatment. And so you'll have an R squared value for each line. So you have 3 R squared values. Okay. Okay. Great. No problem.